My name is Chaney Hart, and my project is Characterization of the Cohesin complex in Neurospora crassa. Cohesin complex is a ring-shaped protein complex that manipulates the structure of the genome. While not fully understood, cohesin, because of its impact on genome structure, also impacts genome functions such as turning some genes on and turning some genes off. Cohesin is implicated in a variety of human diseases, particularly cancer. For example, SA, one of the subunits of cohesin, is one of only 10 genes that is mutated in 4 different types of cancer. So why cohesin impacts some genes but not others is not understood. And a way of looking at this is looking at where in the genome cohesin associates with. This has been done in the past, but there has been an inconsistency in which parts of the genome cohesin interacts with between different model organisms. It's important to understand why this is the case in order to determine how cohesin functions. So to fill in a piece of this puzzle, my project has been to investigate cohesin and characterize it in a model organism in which it has not yet been studied, Neurospora crassa, which is pictured in the bottom left of my poster. Neurospora crassa is a orange filamentous fungus that has been served as a very useful model for genomic structure. So to investigate the relationship between cohesin and the genome of Neurospora crassa, I used ChIP-seq. In ChIP-seq, cohesin is tagged, and an antibody can recognize this tag, and pulling down with this antibody grabs along with it cohesin and all the DNA that was closely associated with cohesin. This DNA is isolated and sequenced, and working backwards from what DNA is sequenced, I'm able to determine what parts of the genome cohesin was closely associated with. So 2 repeatable patterns from ChIP-seq data that I observed was that cohesin on average tends to bind to the very start of genes or to the very end of genes, as well as to the space going past where the gene ends, the 3' end. Another pattern is that cohesin is preferentially associated with convergent genes and is associated with them more than expected. Convergent genes are genes that are transcribed in a convergent orientation. Also in my project, I developed a strain that can be used in further analysis of cohesin in Neurospora crassa. Cohesin cannot be deleted from Neurospora crassa because that organism would not survive. It's an essential gene. But it is possible to introduce a mutation into cohesin subunit RAD21, and with this mutation, this strain is able to grow at room temperature fine, but when shifted to 37 degrees Celsius, cohesin is abrogated, and at this point, the effect of cohesin loss can be studied. So my findings are, the findings that cohesin associates with the end of genes and at convergent genes is consistent with what has been observed in another model organism, fission yeast. So more studies will be needed to determine the implications of these patterns, and a first step could be to see if, despite the fact that these patterns are conserved in Neurospora crassa, to see if cohesin functions differently in Neurospora crassa, such as whether it contributes to genomic structure in a similar way in Neurospora crassa as it does in yeast. I would like to thank the entire Cellcare lab for helping me with this project, and I would also like to thank the Office of the VPRI and the Robert Clark's Honors College for supporting me in this project. Thank you for listening. Have a good day.